Hmm. Assalamu alaikum. Bismillahirrahmanirrahim. I hope you people are ready to attend microbiology class, although we are not right now in sequence. Okay, it was a better topic to have it on Thursday, but unfortunately, I do not have a single hour to spare. Okay, so we will start today our classification, bacterial classification. That's the second chapter, and in history, I request you people, हाँ थर्ड गोल्डन एज पढ़ना था, बिल्कुल पढ़ना था थर्ड गोल्डन एज में कुछ इंपॉर्टेंट चीज़ें, वेरी फ्यू इंपॉर्टेंट discoveries, but I expect you people to go through these. The first one is the most important thing. I told you that genetic engineering products were made available in third golden age. Okay, I gave you two examples of DNA recombinant insulin and DNA recombinant hepatitis B vaccine. Okay, the both of these were available in 1980s, late 1980s, and insulin later on. Uh, as I told you that insulin, there was an issue with insulin before uh, DNA recombinant insulin. Two types of insulin was available. That is one was porcine insulin. Okay, the source was pork, so it was not accepted. Okay, and it was not uh, made available or gain the market in Islamic countries. Okay, one the issue number one. Second was that of second type of insulin was that from beef. Okay, the source was beef. But again, Islamic countries raised an issue that this insulin, that is, this beef, was not halal. Okay, so they did not accept that type of insulin. So it was a very much, we can say, problematic. Okay, issue. So DNA recombinant insulin, it was actually the gene was taken from human pancreas, okay, islet of Langerhans, and it was inserted by genetic engineering in E. coli, Escherichia coli. Okay, E. coli. If you keep in mind, I told you that E. coli is the first bacterial model, experimental model on which all the experiments were carried out, and E. coli has served a lot. Okay, it has, because bacteria have a comparatively small genome if you compare it with viruses. Okay, it is bigger, but if you compare it with higher living organisms from plants or fungi or algae or other animals, it is much much smaller. So it is easier to incorporate a new gene, or it is easier to study the genes of microorganism, or the sequence of genes also, it is easier to study. So that's why E. coli has become an acceptable, or we can say workable model, experimental model in case of genetic engineering. So right now we are getting DNA recombinant insulin from bacteria, that is E. coli. Okay, and other products are also available, like growth hormone. Growth hormone is also produced by the DNA recombinant technology. Okay, another I should say milestone discovery or development in science of microbiology was the development of genomic data. Okay, now we have whole libraries of data of nucleic acids, like sequences of DNA and sequences of RNA of microorganisms available. So if we find a new uh, bacteria, or if we find a new virus, we just have to compare, okay, compare it with available genomic data, or the DNA sequence is match for bacteria, or if it is a virus, then we have to match either the RNA or DNA, whether the type of virus is RNA virus or DNA virus. So we will match, okay, the sequence of the DNA or RNA in order to find out the type or species of that newly discovered microorganism. Then later on it is named, okay. If 80% of the DNA sequence matches to the previous or previously found or already known species, then we will say that it belongs to the same class or same species, okay. But if it is different, okay, that is, this method is also known as DNA fingerprinting, okay, because each and every microorganism have a different DNA sequence. But remember, I told you that bacteria can acquire genes from other bacteria also. So there is a free exchange of DNA, or that is genes, between bacteria, not only from the parents to offspring, but that is horizontal gene transfer, okay, from one neighboring bacteria. Either they acquire genes via conjugation, or that is the transfer of plasmid, okay. The plasmid can be transferred, the genes can be transferred, and there is another method, that is transposition is actually of small DNA sequences known as transposome. Transposomes can acquire different places in actual DNA, okay, that is main chromosome, and they can acquire new places in plasmid also, and via plasmid they can be transferred to other bacteria. So it is very, very difficult in microorganism to we can say, to confine their genes, but it is still bacteria have their distinct, their own fingerprint genes, okay. For example, streptococci remains streptococci, okay. It will not change its shape into staphylococci, or it will not become bacilli. It is because it has got genes which determine its shape. Similarly, biochemical properties, ecological, environmental adaptation. For example, those bacteria which survive in our GIT, they will not be adapted to other environment. For example, they cannot survive on our skin, or in our mouth, or in our pharynx, or in the stomach, because they are adapted. They have genes to survive in that environment. So there are DNA fingerprints, we can say, genes which help us in identifying microorganism. Remember, microorganism do not have such morphological characters like plants. They do not have leaves, they do not have roots or stem, and like animals, they do not have hair or ears or skin or other parts. So it is very difficult to identify them. That's why since 1970 we will learn in classification genomic sequences, RNA and DNA sequences, that is nucleic acid sequences, have become the basis of classification of bacteria, okay. This is a revolutionary, we should say, change, okay, in classification and in studying the taxonomic basis of microorganism. Now microorganisms are classified, and another tool has been developed, that is uh, protein sequence, okay, amino acid sequences of proteins, especially enzymes, because enzymes are very much, we can say, these are actually the survival tool, okay. They are not only important to survive, they for carrying out the main biochemical reactions to live on, but they are, we can say, such an important surviving tool that bacteria cannot in most of the enzyme, they cannot bear to microorganisms, especially they cannot bear to uh, they cannot bear a change, or that is a mutation in those genes which are responsible for the synthesis of or expression of those enzymes which are necessary for the survival. So in that case, amino acid sequences have also become a very important tool in identification, or because we have been able to study the sequences of enzymes or hormones, especially those hormones which are proteins. For example, insulin is a protein, okay, it has got amino acid sequence, and we have been able to study the resemblance in structure of, for example, human insulin and beef insulin. So it has helped us a lot. The sequences of enzyme, we know that the amino acid sequence is based on genes, okay. One gene is responsible for one enzyme. We know that an mRNA plays a very important, a very much, we can say, crucial role in protein synthesis. So in this era, in third era of microbiology, we have been able to find out, or we have been able to develop genomic data, the libraries with genomic data of microorganism, which help us in identification. And we have been also another very important, we can say, revolutionary technique which has been developed during this era is PCR, chain polymerase chain reaction, okay. So this technique has helped us a lot in identification. If we get only one fragment of DNA of a microorganism or any living organism, we can produce a number of copies with the help of this enzyme polymerase, okay. And it has helped a lot, this enzyme, like polymerase and endonuclease. These enzymes have helped us a lot in genetic engineering also, okay. And PCR has become a very important diagnostic tool also, because we have been able to identify with the help of this PCR, we have been able to identify microorganism, especially pathogenic microorganism. So it has become a very important diagnostic tool also, okay. Similarly, we have been in this era in genetic engineering, we have been able to use, and I'm, because I want to stress on those developments which have led us to uh, which have which have been based on microorganism. So we have used in genetic engineering plasmids, okay, plasmids of microorganism as a vector, okay. This is also a very important discovery, and microorganisms have, as I told you, microorganisms have led us to all the understanding of protein synthesis, to all the understanding of mutation. So plasmid has been used as a vector, and there are many other uses also. We have classified plasmids, we have been able to isolate, okay, because we have been able to find out the amino acid sequences, we have been able to isolate with the help of these enzymes, we have been able to isolate those genes which are responsible for antibiotic resistance, okay. This is very very important discovery, because once we know that this gene is found, a particular gene is found in one microorganism, we know that this microorganism would be resistant to many antibiotics. It is possible that one gene can make one bacteria resistant to more than one antibiotics, and the transfer of this gene, that is antibiotic resistant gene, the transfer of this antibiotic resistant gene to other bacterial species, even if they are non-pathogenic, is very much alarming. And these genes, because a lot of work have been done on these genes, a lot of research has been carried out. So these genes have been used as a research tool also in genetic engineering. Okay, because we have markers for these genes, we can use these genes as markers because we have kits available for their identification or their marking. We can find them in a cluster of enzymes or in a pool of microorganism, so we can identify, we can easily locate them. So for this purpose, these antibiotic resistance genes have been used in research also. Another very important discovery in immunology, in case of immunology or in cancer therapies, is that viruses have been research is underway to use viruses for the treatment of cancer. And our body produces, our immune system produces some cytokines are those chemicals which are produced by cells, okay, which are produced by our immune cells and which act upon our body cells. Okay, either these cytokines kill some infected cells, or these cytokines induce the production of some immune cells, or these cytokines have, we have a number of cytokines, okay, produced by our immune system, and we have a number of functions also, but their production is not in our control. One example is interferon. Okay, we have been able to use interferon for the treatment of hepatitis B and hepatitis C. Okay, these interferons are available in form of injections. Okay, and these have been successfully used right now for the treatment of hepatitis B and C. Okay, this is one example, but many of cytokines are under, we can say, clinical trials for the treatment of cancers. And if you as you know, I hope you people know, or you should know, that these trials are first to be carried out in experimental models, okay, in animals. So in animals, first we induce cancer by injecting carcinogenic materials, and then we use these cytokines. But clinical trial means that these have been used in patients. Okay, these now being under a test in patients. These this is a huge discovery that our own human uh, proteins, okay, these are mostly proteins, are used to treat cancers. Okay, these are extracted, and these are, again, these are produced either by DNA recombinant technology, by bacteria. Okay, and these are made available in form of injection, and these must be their safer use must be ensured. Okay, right now you people must have gone through the discoveries of RNA vaccine. RNA vaccine, it is not new. Okay, for us it is new because right now it is available for COVID-19, but uh, research has been under way of more than 10 years back. Okay, subunit vaccine, DNA vaccine, and RNA vaccine were underway, and right now it was, we can say, the research was geared up in order to get a vaccine for COVID. Okay, so it was not an easy task for researchers, but these are all, I must say, important, and I must say, revolutionary. Okay, because before that we used to get, and we still have, for